Hello, my name is Ryan Myers. Thank you for taking a moment to hear about our work on our intranasal cooler for encephalopathy prevention in combat casualties, or as we call it, ICEPICC, a device under development by the company that I work for, Vivonics, for the U.S. Marine Corps. Vivonics is a small business located just outside of Boston, Massachusetts. We focus on pushing clinical solutions outside of the clinic to provide physiological monitoring, advanced diagnostics, and decision support in areas and in situations where it is desperately needed. Areas and situations routinely experienced by our armed forces. This mission of ours is what drives ICEPICC. While ICEPICC is an excellent visual, it is also an apt name for this life-saving device. Seeing the value in developing tools to cool the brain as early as possible, a number of different approaches have been pursued by commercial, military, and academic entities. Unfortunately, these approaches are very civilian hospital focused and lack the flexibility to be brought into civilian EMS, let alone into a military environment. ICEPICC has been designed especially for these non-hospital scenarios, taking lessons from these other approaches to deliver an effective and easy-to-use solution. Cooling of the brain shortly after a traumatic brain injury, or TBI, can minimize swelling and avoid secondary brain damage that can be as fatal as the initial traumatic event. Unfortunately, current methods of intranasal cooling are not adequate for early intervention in military or civilian environments. As you may well know, if you've ever knocked your knee on the coffee table or turned an ankle playing basketball, icing as early as possible is the only way to prevent swelling from occurring. ICEPICC provides a minimally invasive approach to continuously cool the brain in much the same way as the ice pack. The system is highly portable and simple to use for the fastest possible intervention, making it a highly valuable tool to manage traumatic brain injury, as well as other common events that negatively impact brain health like stroke or cardiac arrest. ICEPICC applies chilled, normal air into the nasal cavity using a simple blower to move the air and a solid-state thermoelectric cooler to chill the moving air. This patented approach has allowed us to downsize the system into a portable form factor that lasts over 6 hours on one charge during transportation and can be plugged into a standard wall or generator outlet once a stable role of care has been reached. As you can see here, the cold air is sent from the box through the nasal cannula that guides the air to the appropriate part of the nasal cavity. Currently used for nasal intubation by the military, these nasal cannulas will require limited to no additional training to implement within the DoD. The delivered cold air cools the back of the nasal cavity through convective cooling, and then relies on conductive cooling through the sinuses, as well as convective cooling through the flowing blood to the brain, delivering targeted temperature management of the brain itself. During development, we were able to demonstrate that this means of cooling was able to achieve the same flow rates and exit temperatures as other convective cooling methods trialed in the past by other parties. Multiple publications have shown that maintaining a temperature less than 10 degrees Celsius or 50 degrees Fahrenheit at 25 liters per minute was capable of lowering porcine brain temperature by a clinically relevant level. Optimization and testing of this system is underway through our SBIR Phase 2 with the U.S. Marine Corps. As part of this optimization and testing in Phase 2, we have conducted animal studies, specifically porcine studies, to understand the safety and effectiveness of ICEPICC in an animal model. Our team has maintained cooling for over 4 hours with an average of 2.7 degrees Celsius or about 5 degrees Fahrenheit drop in brain temperature while maintaining normal temperature on the rest of the body. That last part is incredibly important, since current means of reducing brain temperature in a hospital setting are not targeted to the brain itself, and lead to incredible shivering with only a few degrees drop in temperature, leading to difficulties in treating the patient and damage to the rest of the body. Further, cooling of 1.4 degrees Celsius, which would correlate to a reduction of a fever of about 101 back to normal, was achieved in just 14 minutes. Finally, there was no serious damage experienced to the nasal cavity of the animals, and activity was observed to be routine in the 3 days following this cooling event. These studies lead us to believe that ICEPICC brain cooling is safe and effective in a healthy porcine model. Vivonics has been pursuing the studies required to move ICEPICC along a steady TRL pathway toward FDA de novo submission. During our previous meetings with the FDA, they suggested that if the animal studies showed that ICEPICC was both safe and effective, we should proceed to a pilot study in an injured human population. While we are planning to meet again with the FDA to confirm these next steps, we are planning these human studies with our clinical collaborators, and we are applying our additional funding to execute these studies. And being able to jump from this animal study to a pilot study in humans, this path will allow us to get ICEPICC to those in need as quickly as possible. The ICEPICC device is ideally suited for a military environment, holding multiple advantages over other devices under development in this space. Namely, the low size, weight, and power, ruggedized design, will be litter mountable and airworthy. Further, the system uses normal air content instead of including liquids or evaporative chemicals, which can be problematic for various reasons. Finally, the compact design leverages military-approved batteries to power 2 moving parts, making the system adaptable and reliable. We believe, and the data supports, that ICEPICC can be a solution to immediate and unmet need in traumatic brain injury treatment at point of injury and Role 1 care up through higher echelons of care. Not only will this device save lives on the battlefield, but the need is mirrored in civilian America, as well as developed and underdeveloped nations around the world. Our existing engineering team and clinical advisors are working diligently to provide this device to the market as efficiently and as responsibly as possible. Work that has been supported by the FDA during their evaluations of our proposed pathway. In the U.S. alone, there are over 1.7 million traumatic brain injuries annually, over 15% of which are considered severe. While we believe that ICEPICC or versions of ICEPICC can help all 1.7 million, this initial version of ICEPICC has been designed especially for use in severe cases, where a lack of oxygen supply, increased inflammation and intracranial pressure, and increased heating occurs unabated within the first hour following injury. Unfortunately, this population receives targeted temperature management or invasive intervention only after swelling has set in. While the analogy of turning your ankle playing basketball or hitting your knee on a coffee table is a fun way to recall your leg ballooning fairly quickly and not immediately rebounding to a normal size, even with intervention, it also allows us to imagine the more unfortunate realities following a traumatic brain injury. Simply put, the same thing occurs in your head as your leg, but there is nowhere for that swelling to go. Similarly, if we get an ice pack on your brain as soon as possible, we can prevent swelling before it occurs. As mentioned before, the ICEPICC system is highly portable and simple to use for the fastest possible intervention, making it a highly valuable tool to manage traumatic brain injury, as well as other common events that negatively impact brain health, like stroke and cardiac arrest, making the potential impact of this research incredibly high. There is currently no effective solution to treat traumatic brain injury on the front line. We believe that ICEPICC is the answer to that void and are looking for transition partners throughout the military to help fund development through the FDA and field the device once through the FDA. The novelty of the device, however, will require additional funds to get through a pivotal trial, which will be led by our partners at the University of Maryland Shock Trauma Center and University of Texas at Houston, in addition to other sites. Please do not hesitate to reach out to us if you'd like to be involved in the project, or if you'd like to discuss funding or transition opportunities that can help de-risk ICEPICC's development pathway. My name is Ryan Myers, and my direct email and office number are listed here, alongside my colleague Christian Amadio's information. Thank you for your time, and enjoy the rest of the conference.